In this video, you will learn about the product sampling requirements of the California Romaine Testing and Data Analysis Program. The components of the sampling program are sampling stage and timeline, sampling lot size, test organisms, sample size, and sampling methods. We will review each one. The Testing and Data Analysis Program allows sampling at different stages of the product's production cycle, pre-harvest and post-harvest. For pre-harvest sampling, the timeline to test is less than or equal to 10 days from harvest. Post-harvest sampling must be carried out no later than 24 hours after receiving. Sampling lot size is also specified. For pre-harvest, a lot cannot be more than 10 acres, while for post-harvest, the lot size will be defined by the company's post-harvest sampling procedure, but cannot be larger than 10 acres worth of product. The program testing target organisms are Shiga toxin-producing Escherichia coli, STEC, and/or enterohemorrhagic E. coli, EHEC, including E. coli 0157H7 and Salmonella enterica. A molecular or cultural confirmation is acceptable. The sample size is the total mass required per lot to do the analysis. For the program, it must be at least 375 grams. The recommended best practice is to take a minimum of 60 grabs, also known as grab specimens, from the sample area to collect the total mass. The grab specimens are the number of units to collect for the total mass. Simply stated, it is a pinch of material, bits of the romaine lettuce from multiple plants. Grabs must be collected from the edible portion of the plants. Do not pick off just the upper quarter of the leaves. As applicable, include tissue collected from multiple heads or plants. Sample the top, middle, and base of the plant. Sample tender and older leaves. And include the whole leaf and basal petiole. Additionally, when assessing the possibility of contamination via furrow irrigation water, collect leaf samples from beds at the irrigation discharge point of the field, the head row area. Focus on leaves that would contact harvest tools, mechanized harvest equipment, or harvest workers' gloves and apparel. Additionally, samples must be taken using an aseptic sample collection technique to prevent samples becoming contaminated during the sample procedure. The container should be a sterile sampling bag provided by the laboratory, of adequate size for the sample collection. The sampler must wear sterile gloves during the process and must sterilize them with alcohol. Randomize the sampling location and distribute evenly over the entire lot. Walk the lot in a pattern that results in samples inclusive of the entire length and width of the beds. It is recommended to document the specific sample location. Knowing the specific locations can assist with root cause and investigative analysis. Place samples in a cooler and do not freeze the sample. It is a good practice to place a barrier between sample container and the cooling medium to prevent tissue freeze injury. Do not use ice. Use blue ice or ice packs instead. Fill out the chain of custody form properly and accurately with sample collection information and document any deviation. Samples can be taken by a third party as part of the laboratory service, by a sampling service provider, or a company's properly trained employee. Then, the laboratory must process the sample as you have defined it. It is mandatory that the entire weight of the sample is analyzed. Once the results are received, the California LGMA member must upload the data in a special Excel format provided by Western growers that includes the following: acres sampled or cartons sampled, sampling stage, pre-harvest or post-harvest. Note that pre-harvest and post-harvest data will be analyzed separately. Commodity and field pack description. Romaine lettuce, romaine, romaine hearts, and top and tail. Sampling region. For example, District 1, Salinas, Watsonville, San Joaquin Valley, Kern County. District 2, Oxnard, Santa Maria. Or District 3, Blythe, Imperial Valley. Sample date. Organism tested: STEC and/or EHEC, E. coli 0157H7, and Salmonella. Test result: molecular or cultural confirmation for positive or negative results. Although testing is not mandatory for LGMA members, romaine that is currently being tested, such as for customer and/or regulatory requirements, internal company policies, etc., must be submitted as part of the romaine testing and data analysis program. No additional romaine is required to be tested if a handler member does not have a testing policy in place. The data is to be shared on a quarterly basis, a requirement that will be verified during the LGMA audits. This is the end of the video about the romaine testing and data analysis program developed by Food Safety CTS for California LGMA.